This okay? Sound, volume? All right, so thank you, Tina, for the invitation. Thank you all for being here. I'm really excited to spend an hour with you discussing how the future of medicine and how medicine is becoming more and more patient-centric and personalized. So as Tina mentioned, my name is Casey Daniels. I am a postdoc at the National Institutes of Health. I did my PhD at Johns Hopkins in the biochemistry department at the School of Public Health. And now I'm at the National Institute for Allergy and Infectious Diseases, where I focus on sepsis, so the infections of the blood. But today I'm going to talk to you about, again, personalized, precise medicine. So the first question I want to address is, what is precision medicine? Very simply, precision medicine is when the diagnosis and treatment of a patient is personalized or precise based on that patient. And so this idea is not new. Any physician will ask their patients about their family history. They'll ask them about their diet, if they smoke or if they drink. These are all data about the patient that will be incorporated into the diagnosis of the disease for the patient and also an appropriate treatment for them. But the future of precision medicine is a lot more technical. It began with the Human Genome Project, in which the DNA of a human was sequenced. This was more than a decade ago now. And the human DNA tells researchers the map of what that, the genes of a human. And so your DNA sequence will reveal what genes you have and what mutations you have. The genome, though, is modified by what's called the epigenome, and this is the methylation and acetylation sites that exist on DNA, and these methylation patterns can turn on or off certain genes. And so every cell in your body has an entire copy of your genome, but of course every cell is different. And so the epigenome will turn genes off and on specifically within different cells to modify the genome so that it's appropriate for that cell in that situation. So every person's epigenome is unique. The transcriptome is the RNA that is transcribed from the DNA, and of course the epigenome is going to control this transcription from DNA. So this is another aspect, another omics technology, which is called transcriptomic. So we can measure, typically we're measuring the level, the amount of transcript in a person's cells. The transcript can then be translated into the proteome, which is the proteins that are the effectors of the cell. So the DNA is transcribed to the RNA is translated into these proteins, which are the functional units of the cell. These proteins are what transport molecules around cells. They're what provide structure and function to keep your cells functioning properly. So these are the traditional omics technologies, but the metabolome is also a very established omics technology in that we look at the metabolites in your body. These are typically metabolites in the blood or in the urine. We're looking at things like vitamins or metals. And so these, the panel of metabolites that are in your body are your metabolome. And finally, we have the most exciting aspect, I think, of precision medicine, mostly because of how unexplored it is, and that is your microbiome. So every human being has a unique microbiome. These are the bacteria that exist in concert with your human cells. You have more bacterial cells in your body than you do human cells. And these bacterial cells are doing everything from helping you to digest your food to interacting with your immune system to help. There's a constant communication between the bacteria in your body and your immune system. And your bacteria are controlling the way that your white blood cells, for example, respond to new bacteria in your body. They also outcompete pathogenic bacteria. And so you have commensal bacteria all over your body that are keeping the pathogenic bacteria from taking up residence. And so these are the 6 aspects of precision medicine. This is how we can define a patient precisely. We can define your genome, your epigenome, your transcriptome, your proteome, your metabolome, and your microbiome. And we can use the data from all of these omics technologies to diagnose and treat a patient. And ultimately what we're looking for with each of these analyses is we're looking for biomarkers. We're looking for particular genetic mutations. We're looking for an upregulation in a transcript. We're looking for a protein phosphorylation event that will tell us that you belong to a certain category and you should be treated in a certain way. You're going to respond to a drug in a certain way. And these are biomarkers. And every year we're adding and adding biomarkers to our panels. And the pharmaceutical companies love this. And this is the goal of a lot of precision medicine initiatives, is to create archives of biomarkers. Of course, precision medicine is excessive for a lot of applications. We don't need to know your genome in order to tell you that you have the flu. So in any case where you're responding to an external factor, something like the flu or a cold, you're probably, if you have the flu and your neighbor has the flu, you're going to get treated the same way. So in my assessment of precision medicine, there are three major realms of disease that stand to benefit the most from the development of precision medicine. And that is cancer, immunological disorders, and neuropathies, or neurological disorders. So what these three treatment realms have in common is that they are diseases of your body by your body. So in all of these diseases, your cells are going rogue. So in neurological disorders, it's your synapses that are firing when they shouldn't. In immunological disorders, it's your immune cells that are attacking your healthy cells. It doesn't get more personal than an immunological disorder, right? And cancer is what I'm going to focus on today. Cancer is the most well understood disease of the self. So it is a cell, it is a healthy cell that has accumulated genetic mutations. And typically what happens in a body is that you can have a cell. Let me know if I fade out here. Is that better? Better.
Yes.
Typically what happens in a healthy body is you can have a cell that develops genetic mutations. So every time a cell divides, which is happening constantly in our bodies, the entire genome needs to be copied. And we have a pretty, our mechanism for copying DNA is fairly good, but we have a lot of bases. And so there are mutations. And if you get deleterious mutations, typically there are checks and balances, alarms are set off, and the immune system will clear away the sick cells. And what happens in cancer is you get the perfect barrage of mutations, in that you have a cell that is evading all of the checkpoints, but it's dividing constantly, and that's what forms a tumor. So this is the penultimate in terms of a disease of self. And anybody that suffers from cancer, anybody that has cancer, has a unique disease, because this disease is unique as they are. And so again, today I'm going to talk about cancer. I'm going to talk about a heritable form of cancer that's dependent upon the BRCA mutation. But this story actually begins centuries ago. So when a DNA is damaged, when it has a single-stranded break, that means that you got the wrong base in. So G is supposed to pair with C, A is supposed to pair with T, but for some reason A paired with G, and you need to take G out and replace it with T. So you have a single-stranded break because this repair is occurring. What happens in that situation is a process called base excision repair. And it's a very, again, it's an extremely accurate repair of a single-stranded break. If, for some reason, this single-stranded break is not repaired, the single-stranded break will degenerate into a double-stranded break. And this double-stranded break then needs to be repaired by those two processes I described before. A really highly accurate one called homologous recombination, or one that's not very accurate, called non-homologous end joining. What happens in cells that are missing a functional BRCA protein is they don't have homologous recombination, because those proteins are critical for homologous recombination. Now, if you hit these cells with a PARP inhibitor, you lose base excision repair, which means that every single DNA break is going to be repaired by non-homologous end joining, which means every single DNA break is going to result in a mutation. And the cell is going to very quickly reach that point of genomic catastrophe and die. And so if you have cells that are missing BRCA and you treat them with a PARP inhibitor, they die. So again, the pharmaceutical companies are going nuts, right? This is really exciting. This is the first chemotherapeutic that's going to work for patients that have BRCA mutations. So everyone's on board. 2008 was shown in preclinical studies that olaparib, which was the PARP inhibitor that came out of the gate right away, olaparib will shrink tumors in mice that have dysfunctional BRCA. So the preclinical studies are good. 2009, successful phase 1 clinical trial. This is really fast for clinical trials. 2010, successful phase 2 clinical trial. 2011, Sanofi Aventis gets in the game. They do a phase 1, phase 2, phase 3 clinical trial, and they fail. They fail spectacularly. They had a drug called Iniparib, which they claimed was a PARP inhibitor. And what they showed in their phase 3 clinical trial was that it had no effect on patients. It might as well have been placebo. It did nothing. And this is ovarian cancer patients, by the way. And these are all BRCA patients that have BRCA mutations. So Iniparib has a failed PARP inhibitor, and it's the first one to get through a phase 3 clinical trial. And the news was awful. So I joined Johns Hopkins in 2010. I joined my dissertation lab in 2011, and this news came out like 2 months later. I just started my project. And the news was in Nature, it was in the New England Journal of Medicine, it was in BBC, it was in Washington Post, that here was an example of just too much excitement, and this drug class never stood a chance, and we should have never put money in it. And all of the pharmaceutical companies were pulling their trials. Everybody was shutting down. Once a promising cancer treatment, the failure of PARP inhibitors in the clinic may be due to flawed preclinical studies. Go back to the bench, in other words. Well, we did go back to the bench. The field went back to the bench. There were some studies that came out very quickly that showed Iniparib was completely off target. It was not hitting the PARPs at all. Olaparib was on target. So AstraZeneca got back on board. They put Olaparib back into the phase 3 clinical trial, and it was a huge success. So Lynparza is the name for Olaparib, and in 2014, it was FDA approved in the US, EMA approved in Europe for treatment of patients that have ovarian cancer and carry BRCA mutations. And of course, this is in conjunction with the BRCA analysis test for BRCA. So this is a really exciting story of bench to bedside. I think minus the little Iniparib hiccup, this drug went from early bench science in 2005 to a clinical, it was in the clinic being used to help patients less than a decade later. And so this is, again, it's a shout out for basic research that started in the 1950s and now is helping ovarian cancer patients.